everyone, and thank you for joining us today. A warm welcome to the Salk Institute's Power of Science virtual program on conquering pancreatic cancer. My name is Sandy Larakis, and I'm the Senior Director of External Relations and the Salk Cancer Center at the Salk Institute, and I'm very pleased to serve as the moderator for today's program. We thank you for your interest and your support for our Conquering Cancer initiative. Before we begin our program, we would like to share the COVID research that is taking place at Salk. Of course, this is front of mind for all of us, and we wanted to share this. For more information, you can visit this webpage. And as a follow-up to today's program, we will also be sending this link to you, along with resources that Dr. Evans will be sharing about his presentation. We are also recording today's program, and we'll be sending that to you to share with your family and your friends. As part of today's presentation, Zoom has a new feature providing closed captioning, and we've included the how-to slide noted with the yellow arrow. So to enable this feature at the bottom of the screen, you can click, and that will give you a transcript of today's discussion. And today's program is interactive with time for Q&A at the end. We've already received many questions from some of you this past week, and we look forward to answering those. You can still send your questions, and please enter them at the bottom of the screen in the Q&A, and we look forward to answering those. So that we can continue to follow a safe distancing, Dr. Evans is in the trustees room at the Salk Institute, and I'm coming to you from my home. So to lead off today's event, we first wanted to share a message from Dr. Rusty Gage, president of the Salk Institute.
Hello, everybody, and welcome to the Power of Science lecture series. I'm Rusty Gage, professor and president of the Salk Institute, and I want to personally thank you for your interest in Salk research. In this interactive webinar, we'll share exciting advances and cutting-edge discoveries. We're reminded during these sobering times of how critical basic science research is to our health, our community, and the globe. It was the basic scientific research of the Institute's founder, Jonas Salk, that led to the first safe and effective polio vaccine, saving countless lives. At Salk, well-renowned experts in immunology, aging, cancer, plant biology, and other topics are similarly working to tackle some of the most pressing problems of our time. In this lecture, you'll learn more about Professor and Director Ron Evans's important work on pancreatic cancer. Pancreatic cancer is a devastating disease with only 8% of patients surviving more than 5 years. As a part of the Conquering Cancer initiative, Professor Evans has made significant breakthroughs in using a vitamin D derivative in combination with immunotherapy to help combat pancreatic cancer. He will be sharing clinical trial updates on his research. Thank you all for your continued support and interest in the Salk Institute. We look forward to the day we can see you on campus again. Have a good evening.
Thank you, Rusty. And I'm so pleased to introduce our speaker today, Dr. Ron Evans. Dr. Evans received his undergraduate and PhD at UCLA and was a postdoctoral fellow at Rockefeller University. Dr. Evans is director of the Gene Expression Lab at the Salk Institute and holds the March of Dimes Endowed Chair. He is a member of the National Academy of Sciences and the National Academy of Medicine, and is referred to by Google as one of the 50 most influential scientists in the world today. During his 41 years at Salk, he has received numerous awards, most recently this year, the NOMIS Distinguished Scientist and Scholar Award. Dr. Evans and his team are known for their work on genomic networks of health and disease, leading to new therapeutic approaches in both diabetes and cancer. The focus of our program today is pancreatic cancer. He is determined and relentless in these research pursuits, and it's a real pleasure to introduce you to Dr. Evans, a world-renowned scientific leader. Ron?
So along with Rusty, I'm happy to welcome everyone who's out there listening. Really appreciate the time that you're putting into this, and also Sandy for her kind introduction. What I'm going to do today is tell you about some of the work that's going in my lab on pancreatic cancer. This is one of the most feared and deadliest cancers that we know of. And after decades of work on it, it's still considered untouchable. It's highly lethal and poorly understood. Because it's one of the deadliest diseases, if a diagnosis of pancreatic cancer is a crushing diagnosis. And because it's considered untouchable, it is really a sledgehammer when that happens. And so let's think about how we approach this cancer. And if you look here, it is the third leading cause of cancer deaths. And most patients come in with stage 4 diagnosis, that is, it's metastatic. Then there are other forms that are localized in the pancreas and other components that are more complicated about its appearance. Here's why it's one of the most deadliest. Most people come in with metastatic, and the average for that population is 4.5 months. In 1 year, more than 80% of people diagnosed have passed away. So this is a very aggressive cancer, and not only is it feared, but it is just tremendously hard to deal with. It's also, one of the reasons that it's hard to deal with is you can't find it early. And so the nature of early diagnosis for any cancer is key to getting to it and changing the game. But you don't go into your doctor and say, my pancreas hurts. The symptoms are often vague and similar to other kinds of diseases. And so it is this kind of change that doesn't really tell you what's going on until you get the actual diagnosis. One of the interesting features is that in adults that about 85% get some form of rapid onset diabetes. Remember, in the pancreas, part of it is for digesting food, part of it's for controlling sugar through insulin and the islets. My lab studies islets. We study nutrient control and energy control. We study pancreatitis, which is inflammation of the pancreas. It occurs in many people, and it can be lethal, and it can lead to pancreatic cancer. And we study pancreatic cancers. There are 3 diseases of the pancreas that are all tough and important to study. For pancreatic cancer itself, surgical removal is just not available to most people because of the spread of the disease. Less than 20%, 80% can't do it. And interestingly, if you look at the trends for most of the major cancers, prostate, colorectal, and breast, you see that they're going down, as is true for lung. But pancreas is going up. And so this we do not know why it's going against the trend, but it's significantly getting worse. Of course, we all know about Ruth Bader Ginsburg. She was diagnosed actually early in a medical exam, and so she was able to live a long time, perhaps not quite long enough, but it's a terrible loss. And also Alec Trebek. He put up an incredible battle, very positive and very determined, and made it for about 1.5 years, which is actually pretty substantial. But it's not obviously anywhere near good enough. And even with surgery, prognosis is rarely changed. So for this cancer, we have to really rethink. And as I show here, important challenges include improved early detection. We don't have a good way to go. Understanding why therapies fail. You can learn a lot from failure, and that is one of the key things to keep in mind. Identify therapies that are designed for pancreatic cancer. It mostly and only gets therapies designed for other cancers like breast cancer or lung cancer, and they go, let's see if it works on pancreatic cancer, and it doesn't. And think differently. You can't be linear. You can't just say, let's just take a cocktail and see if it works. You have to really have a plan that is a way to go forward and to apply what we think is the most powerful force on earth, which is knowledge. And knowledge comes from the science that we do. That's the most powerful entity. And by thinking differently, I mean change the game. I call this inside out versus outside in. And when you think about how to deconstruct a complicated problem like this, it takes a way of sort of envisioning what your goal is. And if you look at the nature of the tumor and its environment here, the tumor is encased in a living shell of fibrotic cells that are producing inflammatory components, and they're being tied up with the fibrosis. It creates a lot of pressure as that tumor is growing, which excludes blood flow, excludes drugs coming in, and creates this incredible shield or a vault that is protecting this. And this is what we call the stromal barrier. It's a major physical problem, but also it is supplying the energy. It's the fuel supply line for the tumor to grow. And our idea was, if it's a fuel supply line, just like in a military battle, if you cut the supply line, you're going to wipe out the battle. And so we're thinking, how do we reprogram this? And we discovered, using molecular genetic techniques in my lab, that the on-off molecular switch for this stromal barrier is something called the vitamin D receptor. It's the target that controls the function of vitamin D in the body. And as we look at this, because of the way this tumor is designed, the blood vessels get compressed and the T cells come and they try to find the tumor, but instead they find the barrier. And the barrier is normal cells in the body. It's not mutated cells, they're not tumors. So they can't see the tumor. They're around looking, but they can't get there. So we said, can we unlock this barrier and overcome some of these components that have blocked therapy? We're going to try to unlock it by taking advantage of this master regulator, vitamin D receptor, and restoring vitamin D to the system. In this environment, there is no vitamin D. There's an enzyme that breaks down natural vitamin D, so it's completely vitamin D deficient, but that switch is still ready to be turned on. And so when we add a chemical form that is not broken down by the tumor, we can turn the vitamin D receptor back on. And the idea, which is illustrated here, if the tumor is encased in this protective area, can we use the vitamin D receptor to decode the protection mechanism and expose the tumor so that drugs and cells can come in and attack it and change the game. So we start this therapy that I'm illustrating here basically from the outside in. We're not trying to attack the tumor cell first. We're actually modifying the stromal barrier. And when we do that with vitamin D, you see the barrier is broken up. And when that is broken up, what happens is that now cells become available to the outside. And now the T cells can infiltrate, and there they go in. And we see more T cells in patient material that has been treated with vitamin D base therapy. But those T cells are not necessarily active. The way that you activate T cells is with something called checkpoint inhibitors. This is part of immuno-oncology. And these checkpoint inhibitors are antibodies that come in and they turn the T cells on. They lead to the destruction of the tumor. And so this is a process that we are trying to overcome because in the patient population, checkpoint inhibitors have not worked at all in human pancreatic cancer patients. Why have they not worked? Well, because you have not unlocked the entire space or the shield. And so they just bounce off, basically. And so if you want immuno-oncology to work, these powerful checkpoint inhibitors, you have to have a strategy to attack the barrier first. And so with that, we made a proposal to Merck in combination with Stand Up to Cancer to support a trial designed here at Salk in which we would give patients Merck's Keytruda or pembrolizumab. This is one of the major checkpoint inhibitors that's used in oncology, in combination with our paracalcitol vitamin D therapeutic drug. And in a simplified way, I'm just showing you results from that trial. It is an ongoing trial. These are individual patients that are in our trial. And if there's an X, that means that they've passed away. If there's an arrow, that those patients are still going. This is a double-blinded study, so we do not know which patients have received the therapy or not, and neither does Merck, and we will not know that until the trial is completed. Yet you can see here some patients are almost 2 years out. Of course, we're hoping that that's part of our therapy, but whether it is or not, this gives us a lot of information because from each one of these patients, we're going to be getting tumor material that is extracted from them. And each patient has a story to tell. And the story from that patient is usually lost. Most patients, you don't get to really study them in any detail. This is a failure of the system. There's a lot of information, both in failure and in success, and we want to understand how we can use that. So we call this digitizing cancer. This is a way of deconstructing the problem from a very sort of gridded and directed process that we've designed in the lab that allows us to sort of deconstruct this in a specific way. And what I'm showing you here are two needle biopsies. They're really very thin. These are very magnified from two of our patients in this trial. And we're using a special technology. It's relatively new. It's only in about a dozen places around the country. We have one here at Salk that allows us to actually turn these biopsies into colors. We can look at nuclei because they're blue. We can look at the tumor because it's green. We can look at immune cells because they're red. We can look at the fibrotic fibroblasts, which are gray. And you can see them here, for example. And you can see green here. And so we can break this down. We can also look at hundreds of other components in these biopsies in three-dimensional spatial resolution, how these relate to how the cells relate to each other, and also thousands of RNA transcripts. These are products from activated genes that we can look at as well. So from these biopsies, we can extract a huge amount of information that wasn't previously possible. And this is really very valuable. It's, in context of pancreatic cancer, we may be the only ones doing it because the other machines are probably doing other types of things. But we, along with this, developed a novel technology that we call SCAN. And SCAN is selected capture and analysis of nuclei. And the importance of that in this digitizing cancer concept is it allows us to take the biopsy material and take the nuclei from all the cells that are in there, and we sort them in a special type of machine that separates out, because we can identify the cancer cells. Here we're going to say that they're in green, and as they come through the sorter, the green cancer cells get separated from the red stromal cells, and then stromal go here, cancer goes here. Now we can cleanly look at what's happening in the two components when you give the drug and the therapy. And this is key. We've never been able to have this resolution or this ability to actually dig into the details. And this is one patient, and we're going to be doing it with all the patients in our trial. However, Merck doesn't really want that information. That's interesting. They just want to know if their drug works. So it is a little bit surprising. But from that information, we will get a roadmap that will change the way in which we think about this cancer, how it progresses, and how it might be treated. One of the things that, even though Merck is not interested so much, this is how you advance the field, because they're a drug company. They want to develop the drugs. They're not there to deconstruct and digitize the cancer. So we're looking at the genome and all the details of how this cancer gets powered up. Cancer, as it grows, needs energy. And part of this process of the digitalization of this, digitizing this, is to look at the genes in the fuel supply line, that is, these fibroblasts that are making the shield. They are powering up the tumor as it grows. Now, normally, the normal pancreas has a source of energy coming to it, and we just show it as a power grid that comes from a source. We call these super enhancers because we got a special trial, again, from Stand Up to Cancer, on looking at the top 1% of genes in tumor cells or in normal cells that control the power circuit. And we're still running that trial. And through that trial, we can create sort of the nature of that network or that grid. And here's it going to the normal pancreas. What we did is we found that by deconstructing it, there are certain genes in that top 1% that are these genes. You don't have to know their names, but these are 3 of them. And we like these 3 genes for a particular reason. And the reason is because this one stabilizes this particular gene, which is an enzyme, and then this gene acts on the one below here. So one stabilizes that, then this comes here, target that, and that leads to the normal energy flow into the normal pancreas. In the context of cancer, now, instead of having a house, you have like a skyscraper. That's what you have the energy demands of. And so this grid is not enough. So you get an oncogene that we find turns the grid on, ramps it up. This oncogene is called MYC, very common in pancreatic cancer. And now the entire tumor is being lit up. And this is part of the fuel supply line, the energy that is driving this process. And so we then asked, using genetic engineering techniques, can we determine whether these genes are important? This is a scan of the gene in its natural place in the genome. And we went up about 10,000 nucleotides and found a little green region. This is a super enhancer. And we decided to delete it, take it out, using CRISPR technology, which is a genetic engineering tool. This shows it here as scissors, and that takes this off the grid. And so now the source of energy for this is being decreased. And what happens is that the tumor, if normally growing here in yellow, is now down here when we make that SNP. All right? But it's not sufficient to knock it out because the MYC's still getting power, and it's able to power this part down here. So now let's cut out the next part. And what happens when we make that cut? You see what happens again. The tumor really is struggling because it's not getting the power. And we keep going through this entire process with each of the genes. And now you can see here, again, the cut and what happens. And so we are now describing a key energy network that can be genetically engineered to be proven what it's doing. But one of the questions is, can it be drugged? And what I want to say is that that's what we're doing basically, is because one of these genes happens to PRMT1 be the target of a drug that's actually in clinical trials. So we actually got the drug and tested it in our system. And what you see here is this drug simply brings the tumor down to a more than 50% reduction in size, just with this one drug. So now we're not snipping the genes, we're actually giving an orally active drug. It changes the tumor from looking like this here to looking like this here. So we're excited by this approach, giving us entirely new insight into this disease. This is digitizing the cancer, giving us huge amounts of information. Where before we had very little, now we're having abundance, and we have to use machine learning and giant computers to collect all the data that we're getting, but it's really interesting. And just to say what's coming out of a place like Salk, it's basic research, but the impact that we're having in the clinical world is real. We are a small institution, but because we're so focused on what we do, we have a big impact. And this is an example of just the clinical trials and the centers that my lab has been working with. And you can see that they also go all the way around the world. And this has been a great area for us to be in. And I want to just end by mentioning that hope is real. That is the ability to understand this cancer may be now upon us. And so if we can look at this curve here, the survival curve, you saw this before, 4.5 months for stage 4 cancer. I want to show you this is Dr. Stephen Bigelson he's a physician at Rutgers Medical School. He was diagnosed with pancreatic cancer. It's not his area. And knowing that, he knew it was a death sentence. But as a physician, he went to the literature, found our paper, found the trial that we were doing, and prescribed the drugs to himself. And his oncologist gave him our therapy. And what you can see here is since he took the therapy, and from when he started the therapy, he almost immediately started improving. It took about 6 months for all of the signs of his disease to go down. He's now 4 years out and cancer-free. That is, no evidence of disease. And so he's still taking the modified form of the vitamin D as a supplement, basically, and still doing really well. Lots of things are possible. He's one of an example. I get contacted every week by patients who are looking at this, and many that are on trials working with their own physicians. This pancreatic cancer work began in our lab a number of years ago. We were able to get Stand Up to Cancer support, and Jeff Wall and Tony Hunter joined the crew. Danny Engel recently is the 4th person. Michael Downs is a key person in my lab for helping to run this project. Sue Keck is an immunologist, joined us. So that's the 5th lab. The 6th lab is Reuben Shaw, and the 7th is Ed Stice. So it's not just a lab. We have a program that we're developing in this space, and we hope that together we can make a difference. I want to thank our partners, because you cannot get this funded by NIH. They won't fund these speculative programs, but that's what we need at a place like Salk. We're dependent on donors. If you want cutting edge research, you're way ahead of what the funders will give you. So I want to thank the Copley Foundation, the Freeburg Foundation, the Lustgarten Foundation, and Stand Up to Cancer, Wassily, and Pedal the Cause. These are all sources that have really supported our program. It makes a huge difference. We are really on the road. We have momentum. We know what to do and how to do it, and we have the ability to do it. So at that stage, I'm going to stop. Thank you all for listening, joining this call, and I'll be delighted to answer any questions that you might have.
Thank you, Ron. That was great. We have a lot of questions, actually, so I'm glad we have the time to do this. The first question actually is, what's the best way to support this research through donations? And we do have a link on our website for the pancreatic cancer program. And we do have lots of philanthropic needs, especially as we grow this program and expand. And so I don't know, Ron, if you want to take a few minutes to talk about, I know we just thanked the foundations who support your work, but this is definitely a critical time where we need the philanthropic support.
Well, I think that's first, thank you everybody for joining, listening to the presentation. It's my pleasure to do this and to give you a glimpse into some of the work that my lab is doing in this really challenging cancer. And sometimes the more challenging it is, the more focused you get. And we are truly focused on this. And we believe it's going to have a very big impact, the work that we're doing here. And so I am really delighted that you are here listening to us. On the other hand, most of the work that we have done is not from any kind of government support at all. It's through private philanthropy. And I can't tell you how big that impact is. If you want to have cutting edge research, this is really about accelerating the process of getting discoveries. That the government doesn't do that. They're really relatively conservative. And this is all about moving forward at a quick pace, and that requires private support. And I think all of you probably know that the more support you get, the faster you can go. And we want to get there as fast as possible. So it really is a financially driven process. We have the ideas and the way in which we think we can get there. It is sort of thought of as a moonshot sometimes. That's what Joe Biden actually has been heading to get there. But for any kind of cancer, you still have to sort of break through the sound barrier and then be able to get to the stratosphere and then be able to push yourself ever farther. And you need the power to accelerate through that. And that's what we need, I think, for our philanthropy. And the more that we can break that barrier, the faster we can get there. But we are on the heels of this cancer, and we are really close. And so I think it's going to be afraid of us. And whatever we get from that is going to really impact other cancers. So we really encourage, if you can, to support this program. It is really one that we think is going to make a difference.
Great. Thank you, Ron. And I'll plan to send that link along with all the other resources that we've talked about. So let's start with one of the questions. Has research been done in a CHEK2 gene mutation in regards to pancreatic cancer and the family inheritance? So a genetic predisposition.
Well, CHEK2 is a gene that checks cell proliferation, cell division, cell growth. And so when that's mutated, cells have an increased tendency to grow. And so it's commonly mutated in a number of cancers. A study was done at the Mayo Clinic, and out of 96 patients, 2 patients were found to have a CHEK2 mutation. So it is there, but it's not super common. But it's a good question because most cancers collect a number of mutations to be able to become these strong and very lethal ones.
And as a follow-up to that, do you recommend genetic testing for someone with pancreatic cancer to inform other family members? Well,
simple answer is yes. The second statement is absolutely yes. It's been shown through a study that if you get a patient coming in with pancreatic cancer, it was done to then look at as many family members as possible, and significant numbers of family members were found early to get early diagnoses from that. And so this is definitely encouraged. And if someone in your family has it, it's good to communicate with the family and encourage them to get tested.
Okay. Are there any precursors or signals or markers to evaluate before active disease?
That's one of the most important areas to address. There is at least one, which is called CA19-9. This is a type of circulating factor that is actually produced by the tumor. And it is a detectable molecule, and hospitals can measure it, but typically it's only measured as an indicator of therapy during a pancreatic cancer treatment. So by the time it's being used, it's looking at disease that's in place. So yes, you could test in advance, but to do that, we require insurance companies to want to test 300 million people, and they don't want to pay for that. So you could get it done if you want on your own, and you would probably have to do it annually. And so that's something to consider. And it's a very good question. We need other biomarkers that are a little bit easier to test, and we think that that's going to be a key. And there's serious effort at that as part of the program at Salk, including work by Danny Engel, who's very interested in early detection.
Okay. And what is the connection between pancreatic cancer and colon cancer?
Well, first keep in mind the pancreas and the colon are coordinated in what they do. They are physically linked, and they come out of the same developmental lineage. And they're all about processing of nutrients and controlling energy. And so it may not be surprising, they share a number of commonalities in terms of cancer. They also have some things that are relatively unique to each tissue because they're not doing the same thing. For example, the MYC oncogene that I mentioned in the presentation is common to both colon and pancreatic, and both are also relatively resistant to immunotherapy. And so it's a very colon cancer and pancreatic cancers are two of the top three most lethal, and they're very difficult to treat. And typically a diagnosis is a true sledgehammer to a patient. But we have also been working on colon cancer. We had a major paper last year and another one that is coming out of a new approach to thinking about therapy in that disease, something like the vitamin D receptor concept that we mentioned, but it's another closely related receptor with another type of ligand to activate that. And so we're excited about potential therapies for both of these cancers.
And regarding the vitamin D treatment, there's been lots of questions from our attendees. We know that Dr. Bigelson, who you shared, took very large doses, I think maybe through an IV. But for us, as kind of a preventative, how many milligrams would you recommend that we take, since we know that a lot of us are deficient in vitamin D anyway? So what's your advice on that?
Well, whoever asked the question is correct. A lot of people are vitamin D deficient for a lot of reasons. It is an essential vitamin to be taking. It's considered to be sufficiently essential along with vitamin A to be allowed to be fortified into food and cereals and milk and other things. But the reality is we don't get enough of it. The typical average recommendation has been literally 100 to 400 international units, which is, I believe, low. The Endocrine Society recommends 1,500 IUs or international units daily. And I personally take 4,000. And this is not a harmful molecule, but keep in mind, you need to get some sun exposure. So if you couple vitamin D with a little bit of exercise and get a little bit of sun, even 15 minutes, that's a huge impact and benefit to your body. And the other thing to keep in mind is pancreatic cancer patients are very, very deficient when they come in, vitamin D deficient. And the few that are taking supplements actually have a known better outcome. Same is true for COVID. If you were taking supplements with vitamin D before you got the disease, you never go to the ICU. The ones that go to the ICU are very, very low in vitamin D. They're the maximum vitamin D deficiency, and they don't take supplements. And so keep in mind, you can control some of this with daily dosing.
Great. I just wanted to clarify. Thank you for clarifying the very high amounts. And this is something that we can just get over the counter. We don't need a prescription.
Something you get over the counter everywhere. It's
okay.
Relatively cheap.
Okay,
and,
great.
Yeah.
Okay, super. Another question, is there a pre-existing condition like diabetes, which is more prevalent in leading pancreatic cancer?
So say that again.
Is there? So I know you in your former slide you had talked about a rapid onset of diabetes.
Right.
This question is, and perhaps you've already answered it, is there a pre-existing condition like diabetes, which is prevalent for pancreatic cancer? Maybe is there another?
Yes, because that's relatively. There's two parts to that answer. One is one of the leading indications is something called pancreatitis. This is inflammation of the pancreas. This is a tough disease. It's also one that we have worked and published on, and also another one that Danny Engel's lab and ours are interested in, as well as Jeff Wall, actually. And in this disease, inflammation of the pancreas causes great pain and sort of a poor functioning of the pancreas. Over and over again, it also wears down the pancreas, creates scarring, and is a major risk factor for progressing to pancreatic cancer. Pancreatitis is very hard to control, and mostly it's treated with just care and over-the-counter types of therapies. It doesn't have a very good treatment. It can also be lethal, but that is a major risk factor. The other component is there's not a big early component. Diabetes, adult onset rapid diabetes, is a hallmark of many of patients who come in with pancreatic cancer. However, they already have it. So the coordination of the diabetes and the pancreatic cancer means that it's relatively already active. It's still, you can get it earlier if you do get that indication. And it's important to keep in mind that rapid adult onset diabetes should be checked for pancreatic cancer, because sometimes it gets missed and just treated as diabetes.
Mm-hmm. So I know you had covered some, this question is, are there lifestyle factors and changes that improved one's chances of not getting pancreatic cancer? I know you talked about the vitamin D, you talked about sun exposure, you talked about exercise. Are there other things that you would give us as recommendations that we can actively make part of our daily lifestyle in order to prevent this?
Well, I have to say lifestyle is important, and a few things that are challenging in our society is technology, computers. We wind up sitting at our desks, even Zoom. We wind up with technology sitting more. So simply getting up and moving is really important, sort of circulating your blood. Can't tell you how important that is. Even a regular walk or a little bit of a brisk walk, that's a very healthful thing to do. That's not really exercise, but it's getting blood circulating. That really changes the game for risk. And so a little bit of exercise. High-fat diets are obviously a major risk factor. We've been studying, just published a paper on why high-fat diets are risk factors. In fact, the concept of risk is a word. The word risk is actually hides what is really going on. And so you can calculate risk, but it doesn't tell you why that why one thing or another is risk. It doesn't tell you why high-fat diets are a risk or why exercise improves. It may seem logical, but we actually have studied this and had a major paper recently about deconstructing risk from a scientific point of view. And when you understand that, you then can begin to lower risk from scientific strategies as opposed to ways in which we don't fully understand. And so it's a great question, and I am really interested in understanding risk itself from a molecular genetic point of view. It is a real thing, but the number doesn't really help our understanding.
Great, thank you. And just a point of clarification, I know you indicated diabetes as a pre-existing condition. Can you clarify, is that type 1 or type 2 or both?
That's type 2. With pancreatic cancer, it's type 2.
Type 2, okay,
Yeah.
great, thank you. This person asks, how would a person engage in clinical trials? Is that something where, what's the process? I know you shared the slide of working with, health centers all over the world, but if someone contacts you and they've been diagnosed with pancreatic cancer, how would they engage and enroll in a clinical trial?
Well, I get contact all the time, and I have a sort of a couple pages of information on trials, the kind of work that we have, the first things that you should do to begin to put a team together to try to understand what's going on. It's very hard for the patient themselves to understand what's going on because of the fog that you get when you're diagnosed with a major illness. And so you need to get help from others. But trials are out there. We do link people to trials that we're doing. You can go on the internet actually at clinicaltrials.gov and look up trials, and you can type in pancreatic cancer. but it's good to go to get advice at a major cancer center. It is a hospital where there is research going on that is sponsored at the cancer center level, which means this is one of the elite hospitals. And that's where you can begin to get access to new cutting edge therapies and in many cases getting on clinical trials. And all of the major medical centers have trials going on. And that's therefore a good place to be. Many of them are doing different trials. Some of them, like in our cases, even though we're running, saw all the places that we're working with, they're not all running the same trials. We have 4 different trials with our partners. So, and there are different choices to be made depending on the nature of the cancer, how advanced it is, how the shape that you're in, what you can take or not take. And so there's lots of features that come into that. And keep in mind, a trial, you have to qualify. The other part of the trial is you may not know if you're getting the drug or not. And so, but that's the only way that we can progress forward is doing trials. And that's been the fastest way to go for most diseases.
Great, and we do have a question from somebody who was just recently diagnosed with stage 1, and he's interested in hearing more about the physicians that you work with. So I gave him my email address, and we're gonna do that offline to give him those resources.
Yeah, I'm happy to work with anyone to get them connected to the best people.
Thank you.
It's one of the few things that I can do. I know who are the best people, and they're usually very open to talking to patients.
That's very kind of you. Thank you. We'll make sure that we connect with this gentleman with you. And then a question: if you were diagnosed tomorrow, Ron, what is your advice for finding the right treatment?
Well, you have to get good advisors. You have to get a group of people working with you that can first, most commonly, people will want to go to the nearest major hospital to them. And it's been shown that that often is the best in terms of therapy, because if you have to travel or go to another state, it is harder. Now, if you have to travel and go to another state to get something special, you go. And you shouldn't make that to be the barrier. And many people do travel to get therapy, especially if it's only being done in a few places. So that's something to keep in mind. If you're willing to do that, then that opens up a few other extra opportunities. And it's really something that comes down to being personal about the appropriate way to go or not. So there's no right or wrong answer here, but you do want to seek out as fast as you can. This is a rapidly moving disease. You do not want to waste time on treating this. And the way to do that is to assemble a group of people that can help you both find the right clinical center, the right doctors to get in touch with, and move quickly to be evaluated as soon as possible at some of the best places. That's what I would do.
Mm-hmm. Yeah, that's excellent advice. And here in San Diego, Southern California, we're really blessed with many national cancer, NCI cancer centers. But I know some of the people that are on this program today are from all over the country. And so really going on the NCI website or contacting us, because Ron and the whole team here have relationships with other cancer centers, NCI cancer centers. I think that's all the questions that we have. And I know as a follow-up, we're going to be sending the, of course, the link to today's recording, the vitamin D information, the video of Dr. Bickelson's story. And so if there's any other resources that people are interested in, please contact me. Before we wrap up, Ron, do you have any kind of final thoughts?
Well, as I mentioned before, we are really building an incredible program here. We have some of the best talent, and we're in an environment that is known for the kind of state-of-the-art leading research that has translational relevance. And so I do encourage you to think about, if you so wish, to consider a donation to this program. It would go a long way to helping the trials be analyzed, because that's one of the most important ways to get information. And as I've said, there's a lot of information in every patient, and most of that information isn't captured. And this is the key. We're able now to capture this information, and each patient for us is very relevant and very important, but also needs the support to be able to make that into a reality. So you out there can make this program really be significantly better and more impactful by any contribution, because that will have a great impact for all of us. And so, it's greatly appreciated whatever happens, but I understand. It's just I'm happy that you're there to listen to this and see the kind of stuff that's going on at the Salk Institute.
Great. Thank you, Ron. And thank you to everybody on this program. We thank you for your support of the Salk Institute and the power of science. We are reminded more than ever before about the promise and the hope we have in biomedical research and the impact on human health. So thank you all, and take care.
Thank you.